Hi, today on Viva Bites we'll be talking about the formation of crossveins. I'm going on Geoffrey Marcus 2001 paper, The Development and Evolution of Crossveins in Insect Wings. So Marcus developed a framework, a model where the crossveins develop in 3 different stages. We're looking at particularly the Drosophila melanogaster wing. So as a quick reminder, Drosophila melanogaster has 5 longitudinal complete veins, the L1 to the L5, and 2 incomplete veins, the L0 and the L6. Between the L3 and the L4 is the anterior crossvein, and between the L4 and the L5 is the posterior crossvein. There's also the humeral crossvein, but I'm not going to talk about that at all today. So different veins are either located dorsally or ventrally in the epithelium, and this is actually quite important for later on. So as a reminder, the L0, the L1, the L3 and the L5 are all located dorsally. Also dorsally is half of the anterior crossvein, the half that's actually closest to the leading edge. And you also have the distal tip of the L4 that is dorsal. The remainder of the veins, so the L6, the L2, the posterior crossvein and the major part of the L4, they're all located ventrally. So now coming on to the first stage of the model. So Marcus suggests that in the first stage, the number and the placement of crossveins is determined by signalling along the proximal distal axis of the wing. So in particular, it's been shown that genetically CDC42 gene product, which mediates the JUN N-terminal kinase signal transduction pathway, is important to specifying correctly the location of the crossveins. I don't quite know how to pronounce that actually, JUN N-terminal kinase. I've only ever seen it written. The proof of this is that phenotypes of CDC42 mutants show ectopic crossveins at discrete intervals along the proximal distal axis, and the addition of these individual ectopic veins results in shifts in the location of naturally occurring anterior and posterior crossveins with respect to other landmarks on the wing. There's also what appears to be some environmental influence, in particular the sensitivity to temperature. And this was observed, the ectopic crossvein phenotypes have also been observed when flies were heat shocked, and those same flies were defective for heat shock protein 90. So heat shock protein plus defective HSP90 flies at stage 1 results in ectopic crossveins. So overall, the result is that cells at certain positions along the proximal distal axis are specified to signal in the second phase, which I'm now coming on to. So in this phase, the cells that are in the dorsal wing epithelium at these specific positions induce cells on the ventral wing epithelium to take on vein cell fates. And they do this via BMP-like ligands, so BMP signaling. The proof that BMP-like signaling is downstream of location specification lies in manipulations of the DPP and GBB, so decapentaplegic and glass bottom boat. So that they're part of the BMP signaling, and so if you manipulate their levels, this often results in the loss of crossveins. So you can sort of see this as a failure to activate fate in the primed cells. However, there are no reports of such manipulations resulting in the production of ectopic crossveins. So no activation in non-primed cells is one way of interpreting it. Similarly, doubly homozygous mutant flies for a hypermorphic CDC42 mutation, and for CV2 mutations, show only the loss of CV2 phenotype. So this is interesting because, well, normally single homozygotes for CDC42 results in ectopic crossvein, whilst single homozygotes for CV2 results in a crossveinless phenotype. So you've got either sort of, if the order is correct, then you've got ectopic crossveins, so more cells are primed, and then the loss of CV2 would result in none of them or fewer of them showing any crossveins. So this suggests that CV2 is actually downstream of, or perhaps in parallel to, CDC42, as the model predicts. Looking a bit more closely at BMP, how does it set up the crossveins? So decapentaplegic, DPP, which is the bone morphogenetic protein or BMP homolog in the fly, is a long-range signal that specifies the position of posterior crossveins. DPP is found in the adjacent longitudinal veins and forms a heterodimer with glass bottom boat, or GBB. The heterodimer is then moved into the posterior crossvein area by a tetramer composed of short gastrulation, or SOG, and the TSG family member crossveinless CV or TSG2, twisted, I think that's twisted gastrulation. The protease then frees the DBP slash GBB tetramer by cleaving SOG, and this protease is called tolloid-related, TLR, or Tolkien. Knockdown of crossveinless by the RNAi by an RNAi insert results in partial to total absence of the anterior crossvein and complete absence of the posterior crossvein. There are also suggestions that DPP transcription is upregulated by CDC42 activity and the John N-terminal kinase signal transduction pathway. As was the first phase of the model, there also seems to be temperature sensitivity in the second phase, but the result is very slightly different. As if you heat shock flies that are defective in heat shock protein 90 in stage 2, you see a loss of crossveins, but no extra crossveins. So the proof is that losses of crossvein phenocopies are common in populations of heat shock flies. So as a side note, definition of phenocopy is that a phenocopy is a variation in phenotype, generally referring to a single trait, which is caused by environmental conditions, often but not necessarily during the organism's development, such that the organism's phenotype matches a phenotype which is determined by genetic factors. It's not a type of mutation, as it is not hereditary. And finally, coming on to the third stage, so this is a stage where you have the veins appearing properly. The cells of the ventral wing epithelium upregulate Rho and initiate signaling through the EGFR MAPK pathway, so epidermal growth factor receptor MAP kinase signaling transduction. This then specifies the domain of the vein competent cells, and then finally delta notch signaling refines the exact location of the veins within that domain by restricting vein sulfate to those cells in which EGFR MAPK signaling is greatest. These cells ultimately form the posterior crossvein and the ventral portion of the anterior crossvein. Again, as with the other two stages, there seems to be an environmental effect, sensitivity to temperature, as thick vein phenotypes have been observed in association with defective heat shock protein 90. So the result is heat shock plus defective heat shock protein in stage 3 results in thick veins. So in conclusion, by altering components of the 3 stages, insects can independently regulate both the presence or absence, the number and placement, and the thickness and flexibility of crossveins. This capacity for the modulation of crossvein structure in many different ways might have contributed to the evolution of different modes of flight, and allows for a huge number of combinations that can allow an insect to alter the flexural stiffness of the wing. Now, there are parallels between longitudinal veins and crossveins from this model. So, in particular, both longitudinal veins and crossveins require an inductive signal from the dorsal wing epithelium to specify the ventral components. Both require BMP-like signalling in order to form normally, and both also use the EGFR MAPK pathway to specify vein-competent domains of cells, and then further restricts that domain by Notch-delta signaling interactions. So it's possible that over many thousands of years, or millions of years, allelic variation in the components of these signaling processes might have provided the enough variation in flexural stiffness to allow for the evolution of different modes of flights in insects. And that's all for today.